Okay, so we're back here to talk about transport across a cell membrane, which is section 3.5 of your book. It starts off with passive transport. So transport across the cell membrane. Remember, cell membranes we need to allow some transport, but not everything should transport in there. So if a cell membrane lets some things in and not everything in, if it's sort of picky and choosy, if you will, then we would say that membrane is selective, because it's selective about what comes in and what comes out. Permeable is what's going to cross. How permeable something is means how easy it is for something to be transported. So if a membrane is selectively permeable, that means it lets some things in, but not everything in. All right? So, and that's exactly what we know we need to have happen, right? The reason why a membrane is selectively permeable is because of that phospholipid bilayer, right? Things that are fat soluble, they're going to come in real easily. So fat soluble substances like our fat soluble vitamins, vitamin D, for example, K, A, D, E, and K are our fat soluble vitamins. Those are able to get easy access to across our cell membranes. So are drugs that are fat soluble. So if you're in the profession of drug design and you want a drug that's going to get like all the way into the brain pretty easily, it's going to need to be fat soluble because it's going to cross through all sorts of cell membranes. Things that are polar are not going to easily pass the charge. They're going to be repelled by the cell membrane. Things that are water soluble are not going to easily pass. So ions, charged particles, are going to have a harder time coming across without some help. So because the membrane doesn't let everything in, but it lets some things in, we say that it's selectively permeable. So the question is, what's the criteria for the transport across the cell membrane? Some things are going to go, some things are going to maybe go, some things are not going to go at all. So the big criteria is what I just said, lipid solubility. Something that's soluble in lipid will easily pass. The next question we're going to want to know when we're talking about transport across a membrane is whether or not it requires energy to transport something. So something that's lipid soluble traditionally does not need energy because it just diffuses right through, right? But sometimes we need to actually use some energy to do it, and that is a little bit of a different story. So this would be an example of no energy required. So on the bottom of the slide there, you see there's like a cup of hot water. You can't see that it's hot, but I'm telling you it's hot. So you boil some water, and then you plop in your tea bag. The tea bag, of course, is encased in a membrane, right, in this picture. So that, and remember when the temperature is high, molecules are moving around really fast. So the molecules in that hot water are moving all over the place. They're bumping into that tea bag, and the stuff in the tea is going to then just diffuse right out of the bag. The bag is permeable to the stuff in the bag. And also to water. So water's going to come into the bag. The tea's going to come out of the bag. There's no energy required. You don't have to do anything there. You know, you could put, and you've all done this, I'm sure, you could just put tea in a room temperature water, and ultimately it will come out of the bag. It's going to take longer. The warmer the water, the faster it's going to happen, right? But you don't have to actually do anything extra energetically to get the tea out of the bag. And so when you have a lot of something in one place and not a lot of it someplace else, we say that we have a concentration gradient. So in this case, there's a lot of tea in the bag, and there's no tea out of the bag before the bag goes in there. So the concentration gradient for tea is it's high inside of the bag, there's a lot of it in the bag, and it's low outside of the bag. So with transport, that does not require energy. It does require that you have a concentration gradient. So particles are always going to move from where there's a lot of them to where there's a little of them. That's a universal law, right? So when you have a different concentration, so when you have a defined physical space in which there's a concentration inequity between two substances, right? You have a lot of it, or you have a lot of the substance someplace and not a lot someplace else. That's a concentration gradient. So the particle will always move from high concentration to low. And that's just that's just a universal law. It's a law of nature, if you will. So when there's no energy required in regards to transport, we refer to that as passive transport. It's a naturally occurring phenomenon that does not require the cell to use any energy to accomplish the movement. So in order for passive transport to occur, you do have to have, across a cell membrane, a couple criteria do have to be met. The first is the membrane has to be permeable to the thing you're trying to transport. It's got to be able to get in there somehow. The second criteria is there has to be a concentration gradient. So I'm going to say that again. For passive transport to occur across a membrane, the criteria for transport would be 1, the membrane has to be permeable to said thing you're trying to transport. So the membrane must be permeable, and there has to be a concentration gradient for that substance. So the membrane must be permeable to that substance, and there has to be a concentration gradient for that substance, which means there has to be more of it on one side of the membrane than on the other. So if you look at this picture on the left here, this is just standard passive transport, doesn't require anything other than the concentration gradient. So when you have this kind of a setup, we call this diffusion. So this is how fat-soluble substances easily pass. You've got a lot of them on the outside of the cell, not very many of them on the inside of the cell. They're going to move from high concentration to low because the membrane is permeable. So the first criteria, membrane permeability, check. You see it going right through. Second criteria, concentration gradient. High concentration here, low here. It's going to move from high to low, right? That's called diffusion. There is no energy requirement. So you could write diffusion, no energy. The thing that it does require is a concentration gradient and a permeable membrane. Another example of passive transport, which does not require does not require energy, is the transportation of water. So these cute little Mickey Mouse guys, these are water molecules. So water will passively transport across the membrane because water is really small. Yes, it's polar, and yes, it's water, but there's pores. There's little channels in our cell membranes for water. This is one of them right here. They're called aquaporins, and they're proteins. So water will move from high concentration to low concentration, just like another solute would. So water will move. In this case, the water looks like it's going from high concentration here out this way. So the water is going from the inside to the outside. It could do either way. It just sort of depends on the concentration of water. The transportation of water across a membrane is referred to osmosis. So diffusion and osmosis are basically the same thing, but osmosis refers specifically to the transport of water, and diffusion is anything other than water. And then the last one that can be a little bit confusing is over here on the side, and this one's called facilitated diffusion. And the reason why it's called facilitated is because we need a little help, but we do not require energy. So the little help here is this transport protein. And notice, so we got this, which will help it. So the membrane in and of itself would not be permeable to these green things, but we've got this protein here that does allow it, and it's just open. So we have the permeability thing handled, and then the other thing is, do we have a concentration gradient? Well, look, we've got all these green hexagon things over here and not in here. So these guys are going to want to come inside, right? They're going to move down the concentration gradient from high concentration to low. And that is called facilitated diffusion, right? So you see them coming in there. Whoops. Right. So summary here, 3 types of passive transport. They're all passive, which means, write this down somewhere, that means no energy is required for any of them. In all cases, we have a concentration gradient, high to low. In this case, it's lipid soluble diffusion, right? In this case, we need a we need a facilitated protein, but it's not requiring energy. It's still high concentration to low, and osmosis is specifically the movement of water. All three of those are terms to describe passive transport. All three of them rely on a concentration gradient, and all three of them rely on the ability of that thing to be able to be transported. So the membrane has to be permeable to those things, even if that permeability requires some assistance, like in the case of facilitated diffusion. What you don't see happening here is energy. So I'm going to show you what the energy one would look like. So the other type of transport is the kind that requires energy, and that is called active transport. So the characteristic of active transport is, big characteristic is it requires energy. That's the big thing to remember. But the other thing about active transport is we are pumping generally against the gradient. So you're pumping something towards the high concentration as opposed to from high concentration to low. So it's like swimming upstream. So that's an analogy that maybe some of you can sort of relate to. If you've ever been inner tubing in a place where you have a creek or a stream that's moving. So, if you just hop in your tube and you float downstream, obviously that doesn't take any energy at all. You're going with the current, which is like going down the concentration gradient. However, when you decide you have to swim back to your shoes, now you're going against the gradient. That takes a heck of a lot of energy, right? That's really hard to do. So that's active transport. It's swimming upstream. And so the same kind of a thing with active transport in a cell, you're pumping a solute or a substance against the gradient. You're pumping it upstream, and it requires a lot of energy for that to happen. The classic example of active transport in the body is a thing called the sodium-potassium pump, which requires ATP. So this guy right here could be a sodium-potassium pump, right? And you can see animations of these if you want to. But basically, what's going to happen is these guys come in, they bind to sites, and then we have to take, with the sodium-potassium pump, basically 3 sodiums move into the site, and then 2 potassiums get on the other side, and then we pump them against the gradient. You pump the sodium out against the concentration of sodium, and you pump the potassium in against the concentration of potassium, so it requires energy. And that energy, always, all energy in the body, every single time, is ATP. So active transport requires ATP. That's it. It's not free, right? Passive transport is free. You don't have to apply any additional energy. The energy is kind of embedded, if you will, for the lack of a better term, embedded or kind of included in the concentration gradient. But you don't have to add any more energy to it. Whereas with active transport, we need to require energy. Okay, that's transport. So one last thing I want to mention is, in the same vein of getting things into and out of the cell, sometimes the cell membrane itself can kind of engulf or spit out certain substances. And so the term for that is when the cell's kind of engulfing something and bringing it in, it's called endocytosis. So what you see here, so this would be the cell membrane, and basically the cell membrane kind of envelops itself. The thing kind of, whatever you're trying to bring in, sort of comes around the outside of the cell, and the cell membrane kind of grabs it and sucks it in. So this would be the inside of the cell. That's called endocytosis. It's bringing it in. Now, if the cell's packaging stuff up inside the cell, neurotransmitters or chemicals or whatever, we make them inside of the cell, then we pack them up in this cute little vesicle, this little membrane sac, and then that vesicle goes down and it fuses with our cell membrane, and then those little neurochemicals, if in this case, get spit out. That's getting spit out of a cell, being exported, is called exocytosis. So when your cell is, sometimes they refer to this as cell eating. So when it's eating something, it's called endocytosis if it's something solid. They also refer to it as pinocytosis if it's something liquid, but in both cases it's endocytosis. It's bringing something into the cell. Exocytosis is getting something, exporting something that you've made in the cell out. So endo and exocytosis. Okay, so I'm going to stop there, and then I'm going to come back to introduce you to some organelles, and that will be the last part of this. And then you're going to watch a Bozeman video on cell organelles and transport. All right, I'll see you in a little bit.